All right, welcome back to Plant Science 1. This week we're talking about leaves and continuing our comparison between monocots and dicots. Before I get into it, I'm going to do a few review slides just to make sure that we're all tracking. So just a few questions, I'll ask them, and then you can share them with your cat if you want. Um, so what are we looking at here? What is this? Is this a monocot stem or a dicot stem? And the answer is monocot stem because the vascular bundles are scattered throughout the entire width of the stem. They're not organized into rings or bundles in a perimeter only. So monocot. And of course, by subtraction, this is a dicot. So my next question is, where are the xylem and phloem located? Right? Are they located inside or outside? So the best way to remember this, especially for dicots, is to think, okay, in a tree, if you cut it, the inside's wood, which is always xylem. And so the phloem must be on the outside. So phloem on the outside, xylem on the inside. So this is a monocot. All right. Yeah, repetition's a good teacher. So here we have a pith in the center. We've got vascular bundles when it's immature. As the dicot matures, those bundles merge into what we call a cambium. And then, of course, outside of that, eventually you get a cork cambium, which starts to produce bark. One more time, this is a monocot. Now, while we're looking at it, which way do the phloem and the xylem orient? Well, ironically, the best way to remember is to think of it just like a dicot. So the xylem are always towards the middle and the phloem are always to the outside. So that, if you're trying to remember for monocots, just think it's no different than a dicot. Yeah, this slide doesn't really show you much, but yeah, cork cambium uh, is, there's no arrow. I'm not sure what this is pointing at, but it's a good slide. If I get my pen out here and draw something. So let's highlight the cork cambium. Well, here's an interesting thing. Vascular bundle. We have xylem here. I'll give it an X. And we have phloem here. So we have xylem and phloem. Then between them must be the vascular cambium. In this case, not so much a bundle. It's bundle-like because this is a bit grouped. This is sort of an immature stem, somewhere between, early growth, young growth. So the phloem is not formed like the continuous mesh. Now, the cork cambium would be outside of this, so typically that would be around here. And that's pushing out cork to the sides, which splits as the tree grows in width, forming cracks. And if it's a soft bark, it may stretch like a beech tree or something like that. So, a little closer look. I wish I'd kept that up. I would have known what was coming next. Um, yeah, just a closer look at that. Just another look at wood. You're going to learn a lot more about wood from Wayne, so I don't want to get too lost here. But again, we're talking about mature dicots. So we have xylem and we have vascular cambiums, we have phloem on the outside, then we have cork cambiums, and we have cork or bark. So what are we looking at here? Vascular bundles. Is it a monocot or is it a dicot? Now, in this case, it's difficult to determine if I just show a picture like this. It might be because you don't know if it's in a ring. Where here we can see it's in a ring. But if you remember from my lesson earlier, that the monocot tends to have a face. So it's got like big eyes, a big kind of mouth or nose, with phloem in the head, in the forehead. So we've got xylem down here, and then there's these larger rings separating them. That's kind of how you tell if it's a monocot. If you're looking just at bundles alone, how often that's going to happen in your horticultural lives, I don't know. And here you go. Monocot or dicot. I gave it away. So it almost looks like a Halloween picture, right? Like this, like a skeleton with the scary grin. Anyways, okay, monocot. I think we've hammer this down. And the main function of phloem in a vascular bundle is what? To transport water, to transport sugars and amino acids. So its principal function is to transport sugars and amino acids. You could also add plant hormones to that. And for xylem, of course, it is the opposite, which is principally to transport water. So let's move on to leaves. Found these pictures on the web, tried to find royalty-free ones. I don't know who took this one, but pretty picture. Anyways, leaves are obviously an important part to plants. You know, they make all the sugars. They basically are solar panels, you know, generating electricity. And in many ways, they really are like solar panels because they take sunlight and they liberate electrons from water, right? And they use carbon dioxide and these electrons and stitch them together to make complex carbon molecules known as carbohydrates, or as we know as sugars. And of course, a byproduct is oxygen, which, you know, is obviously beneficial for us. So that's the gist of photosynthesis. Um, in your labs, you'll be able to hopefully watch the gases change in plants as they undergo photosynthesis, but that's at a later date when we look at photosynthesis in a little more detail. So again, I don't want to get too lost in this. But as far as leaves are concerned, a critical aspect is obviously getting carbon dioxide into the leaves and oxygen out of the leaves. And plants do that through what are called stomata. You may have heard about these before. These are little pores on the leaves that can open and close, just like the garage door in a house, letting stuff in or out and controlling access. And of course, they permit the exchange of gas, in this case, letting carbon dioxide in and O2 out, where the xylem, which we discussed last week, delivers the water. Because remember, we need water. We need carbon dioxide, we need water, we need electrons. We need all these things in order to make sugars. So we have xylem delivering water to the leaves, and we have stomata opening and closing, letting gases into the leaves. Some more pictures to show the same thing. I'm just showing some sap. If you cut a twig and there's any kind of root pressure, it just shows that the sap will leak. This is obviously the same principle as tapping a tree to make maple syrup, right? The phloem pressure is delivering sugars, which we boil down to make maple syrup. So now, as far as the transport is, again, from last week, we talked about how sugars are transported to areas of need. We call those sinks, in this case, an apple, right? Other areas of needs could be meristems, where there's mitosis happening, developing fruits, storage, as in a potato, or maybe a flower. So we have to be mindful of that source-sink relationship. And the leaf is the source. That's where it all starts. And so when we're talking about leaves and their anatomy, it's important to remember that we're talking about the source for everything that happens in the leaf. So going back to the stomata and understanding how they work, they are openings, right, that inflate and deflate. So in this particular case, the stomata is deflated, and you'll notice that there's not much of a opening. In fact, that line there is a solid layer of tissues that's on the inside of each facing guard cell. So there's a cell on one side. It's maybe easier to see here. There's a cell on one side, and there's a cell on the other side. And then they have sort of a stiffened side that faces each other. And it's stiff so that when the cells inflate, that side stays in place and sort of warps. And that way the gases can kind of go in and out. The best example would be to look at two balloons. If you tied two balloons end to end, they're fixed at this point. As you inflate them, a pore opens up, right? And it's here where the stomatal pore allows gases in and out. So how they do that is by pumping potassium into the stomatas. So potassium is pumped into the guard cell, into the guard cell, not the stomata. Potassium is pumped in, which increases the osmotic potential of that cell. And with increased osmotic potential, water moves from the neighboring cells into the guard cell, causing it to enlarge, which opens the pore. So the plant can control whether the stomata are opening and closing based on levels of potassium. Um, Interestingly enough, the idea that CO2 entering and exiting is controlled by the plant alone is a little bit simplistic, because there's situations where plants can run out of water. So if they're water stressed, if a plant is limp, does it have enough water to move into the guard cells? No. So sometimes the fact that it's relying on water pressure to open the guard cells will also help prevent the leaf from losing water in cases of extreme stress, because the guard cells stay stuck shut, and that prevents water vapor from evaporating from the center of the leaf. So at a later time, you may look into something called stomatal conductance, where we measure how much air is moving in and out of stomata. Now, so we know that potassium is required to move into the guard cells, right? So potassium levels increase in both sides, and that causes water to flow into the guard cell, causing it to inflate, which opens a pore, letting oxygen out and CO2 in, right? Now, in many respects, potassium doesn't actually play a lot of significant roles in plants physiologically in terms of chemical reactions, but it is a very, very important nutrient. In fact, a lot of the times, fertilizers have to deliver more potassium than any other nutrient. And so I asked the question, looking at this big tree, how much potassium do you think it needs to operate all the stomata on its leaves? Obviously a lot, right? So this is why potassium is such an important nutrient for plants. If they run out of potassium, they can't open their stomata. If they can't open their stomata, they can't undergo photosynthesis. If they can't undergo photosynthesis, they don't grow. So potassium is an incredibly important nutrient. Um, Typically what we like to say is that potassium is responsible for most things related to osmosis. So they help move water and to fill things and stuff like that. So the stomata is obviously the most key one, but there's some other roles. So let's look at leaves in terms of their anatomy. So we've looked at, their stomatal function and what the leaves do. So let's look at the different leaves. Continuing with the theme of dicots versus monocots, uh, obviously monocots are grasses, uh, longer leaves, where dicots are non-parallel veined leaves or broad leaves. They come in lots of different shapes and sizes. Monocot leaves do to some extent as well, but the general gist is that the leaf veins would be your best indicator of whether it's a monocot or a dicot. So how the veins, which are really vascular bundles, are arranged in the leaves. And for monocots, we talk about parallel venation. So the veins are parallel. And for dicots, we call it reticulated. So they branch, right? They form contributory, like smaller little veins that combine into larger ones, which then combine into the main one that heads to the trunk. So that's the key difference in terms of the large sort of macro anatomy of monocots versus dicots. If we start looking at the more intricate levels of dicots and monocots, there's a couple of differences that we should really be aware of. This is showing the upper epidermis of a leaf. We've got a little trichome. We have the epidermal cells. But actually right above them, this thin layer here is a waxy layer we call the cuticle. Plants have different levels of cuticle. Some are very thick, some are very thin. They are sort of meant to seal off the epidermis to minimize water loss, because the plants want to be able to control water loss through their stomata. They don't want to risk losing it through their leaves. But all plants are different. You look at a cucumber or any kind of melon, they have almost no cuticle, and their leaves are very, very paper thin and soft. and they have huge water needs because they evaporate so readily. Whereas you look at a cactus, and the cuticle is incredibly thick because it's dealing with a very dry, extreme climate. Now, below that is something called palisade cells. Let's look at that. These cells down here, these are sort of parallel, tightly packed, tall cells. Now you'll see that they have chlorophyll in them, so they're chloroplasts. So they do undergo photosynthesis, um, but they are tightly packed. So I always like to think of the way to help you remember them is if you think of palaces have columns. So palisade cells are arranged like columns, and they're just underneath the top or the upper epidermis to give it some strength. And then below that, we have what's called the spongy mesophyll. So you'll notice there's lots of air spaces in here. And that's to facilitate gases moving through the leaf. Because as we talked with initially with stomata, the whole point of the leaf is to undergo photosynthesis. And one of the main ingredients for photosynthesis is carbon dioxide, which means you need to get gas into the leaf. So the stomata, which you see here, cut in half, is letting gases in, and they diffuse throughout the mesophyll, or what we call the spongy mesophyll, and that lets the gases get to the surface of every single spongy mesophyll cell so that it can undergo some pretty intense photosynthesis. Instead of looking at diagrams, let's look at some cross sections. So this is a dicot leaf. The UE is the upper epidermis. There's actually a thin cuticle above it. And here you see the palisade cells. And then below that is the spongy mesophyll. Here's another cross section showing the upper epidermis. That was terrible. The upper epidermis, and then palisade cells here. And then your spongy, really spongy mesophyll here. That really shows how much space is inside the leaf so that gases can freely get in and out, which is hugely important. This slide, it's really difficult. I think in your labs, this is something you should look for. You should be able to see them in your cross sections. These are vascular bundles here. And if remember, we started off with the leaves by talking about how monocots have parallel veins and dicots have reticulated veins. And when we're looking at a vascular bundle, that's actually a vein cut sideways, so cut across. So this would be a vein traveling through the leaf like that, and we've cut through it, and we're looking across the vein. So that's a vascular bundle. And the xylem is located at the top, phloem at the bottom. Which kind of makes sense because a lot of the sugars are being made around the spongy mesophyll. And so the sugars need to be able to access the phloem. So its proximity is close to the spongy mesophyll. And of course, you've learned last class that wherever there's phloem, there is xylem for the very simple reason of moving the sap. And then, of course, the main vein in the center of the cross section, you can see the large xylem in the top and then the finer phloem below it. So these things, they would branch and connect and contribute to the central phloem that takes the sugars away from the leaf to an area of need, or a sink. So going back to the anatomy, I asked the question, to palisade or not? So dicots have palisade cells. Remember I said a pallis has columns? That's those column-like cells that are at the top of the leaf under the epidermis. If you look at this picture of this hibiscus, you'll see that the leaves are sitting out horizontally, right? Imagine if you're holding a big sheet of plywood from the side. If it was really soft, it would just sag, but because it has some structure, the plywood sheet extends out horizontally. And the palisade cells do that. They stiffen the leaves so that they can stretch out sideways like solar panels and collect the sunlight. Now, one of the key differences between monocots and dicots is that monocots do not have palisade cells. The leaves are typically oriented vertically. Think of the grass on a lawn in turf, or think of a daylily. The leaves are sort of narrow, they have parallel veins, and they're vertically oriented. They may arc over, but generally they are vertically oriented. So they don't need the extra strength of the palisade to keep them from sagging. And if you think about it, the palisade cells, because they're not as perfused with gas, they're not as productive in photosynthesis as the spongy mesophyll. So a monocot does away with the palisade because it doesn't need it, and it has just spongy mesophyll. So there's actually a really good gas exchange, and they're a little more efficient with their photosynthesis. So where it may look like a monocot has less leaf area, it does make up for it in terms of its efficiency with gas perfusion. So another important difference, and this is often overlooked, is that in dicots, the stomata are typically underneath the leaf alone, where in monocots, they're on both sides. Now, think about it. This leaf is oriented vertically, so gases can access both sides freely, right? And if the sun is shining, typically it's going to come at a steeper angle, so there's less drying pressure, that sort of thing. If you look at a dicot, it's a bit of a different story, right? Like the sun is impacting on the surface of the leaf directly. The wind is blowing over the leaf directly. And more importantly, when it's wet, water coats the entire surface of the leaf. And so dicots typically have the stomata only on the underside, the underneath side. You'll see that in your slides when you look in the lab. And that's so the stomata are protected from the increased evaporation pressure of the sun and from airflow and from rain plugging up the pores. So here's a cross section of a monocot leaf, and we see that they still have a cuticle on the outer surface. They have the epidermal layer, just like a dicot, and they have a vascular bundle because they have veins. Remember, these are parallel, and they're parallel. Does that mean that this vascular bundle is going to connect to the one beside it? Not in the leaf. No, they actually follow each other parallel all the way to the base where they attach to the stem, and then they join the stem's phloem. So they're not branching, they're parallel. And of course, there is an up and a down, which will become more obvious when you go to the next cell. But the xylem is near at the top and the phloem is at the bottom. So that is conserved across monocots and dicots. You'll notice there is another parenchyma cell here. We talked about parenchyma. Remember I said when in doubt, say parenchyma. But this ring of cells that surrounds the xylem and phloem is called a bundle sheath, and that is with parenchyma cells. Okay, so looking at a monocot leaf, we're looking at corn, or ZMAs. That should be italicized. Um, and we have guard cells. This is actually at the top. Doesn't matter because it's a monocot. Guard cells. And here's the pore, right? This is where gases are going to go in and out of the leaf. And there's often an empty space below the pore just to make sure that gases can get in and move instead of having it plugged up right away. Let me clean that up. It's kind of a lot of stuff drawn there. So enough of that. What's interesting here is something called the bulliform cell. These things operate in the same principle as a guard cell. So here we have a corn cell, a corn leaf again. So there's a guard cell and a guard cell, a stomata. Here's the opening underneath the stomatal pore. And at the bottom, guard cells, pore, open space, vascular bundles. Now, the bulliform cells are really neat because what they do is potassium, once again, potassium, is pumped into these cells or pumped out. And so if potassium is pumped out, they shrink because water leaves. So they collapse. And by collapsing, they actually fold the leaf up. And that helps to minimize water loss through a number of means, basically changing the orientation of the leaf. My next slide probably shows it a little better. Let me just move to the next slide. So on this slide, I've got the bulliform cells shrunk a bit up here, a little smaller, but there they are. And my simple analogy here, forgive me, is an accordion, right? So you can see like these are like the bellows in the accordion. As they shrink, they would be like pulling the accordion together. So that's what the bulliform cells. And if the leaf, if it was normally flat like this, it folds up into sort of a V shape horizontally. Well, in a vertical V, think of it like that. So you'd be staring straight down into this bullseye, looking from the top down to the bottom of the leaf, if that helps depict what I'm trying to say. Um, so bulliform cells help fold the leaf, and by folding it, here you can see how extreme the fold is. It's more difficult for air to swirl and remove moisture from the leaf. So it's a water stress response. The other thing that happens is it changes the angle that the sun impacts the leaf. So let's say the sun is shining here and shining away, and it's impacting this leaf. It's going to hit the surface of the leaf at a more acute angle. Whereas when the leaf is folded up, the sun is going to have a little more difficulty accessing the center of the leaf, which reduces just the temperature, which reduces the amount of evaporation. So again, it's a means for dealing with water stress. And what's interesting is, again, with water stress, if you don't have enough water, these cells collapse. So if you're looking at turf and you want to know if it's water stressed or not, I mean, usually it's pretty obvious. We're just judging by how hard the ground is. But you can look at the leaf blades, and if they're folded lengthwise like this, then they're water stressed. If they're more open, they're less water stressed. So some review. A dicot leaf has stomata primarily on the bottom, has spongy mesophyll, has palisade cells, has an upper and a lower epidermis, and has a cuticle, and has a vascular bundle with xylem at the top and phloem at the bottom. Okay? Monocot leaves, pretty much identical except stomata are on both sides, and there are no palisade cells. And they have the addition of bulliform cells. That's the key anatomical differences that we're going to be exploring as far as monocots and dicots are concerned. Let's slide into gymnosperms because there's some interesting differences with the inside of a needle in an evergreen, right? So evergreens are exactly that. They're evergreen, with few exceptions. Like a larch might, it drops its needles, but these needles, they stay on all winter, which is an extreme condition. It's cold, it's windy, it's drier in the winter. So needles are very well adapted to control evaporation from them. This is a Scots pine, Pinus sylvestris. Oh, look that, there's Sylvester. So in a gymnosperm needle, one of the key adaptations is something called the hypodermis. Okay, now it's not hypodermic like a needle, it's hypodermis. We still have an epidermis, so we have an epidermis, which is the outermost layer, which, by the way, is still coated by a waxy cuticle, a thick waxy cuticle. So that's your first layer of moisture defense. And then below that is the hypodermis. I've zoomed in on it here, and you can see these tightly packed, thick cell walls that are underneath the epidermis. They create the hypodermis. And this is all about preventing water from diffusing through the cell walls to the exterior of the needle and then evaporating into the atmosphere. Remember, cell walls are made of cellulose principally, which is just like paper towel, which if you drop paper towel in a puddle, easily wicks. So if, like in the spongy mesophyll, water can easily move between the cells through the cellulose, no problem, just like in a paper towel. But these cells have thickened walls. There's a chemical pumped into the cellulose, often called lignin or suberin, which prevents water from passing through the cell wall. So there's no way for the water to get through, so it controls water loss. The only way water can exit is through the stomata, which is yet another adaptation. The stomata is recessed. So let me highlight this, recessed. So here are your guard cells. And then we have a deep, deep passage to get all the way down into the needle to where there's that air chamber. And then your mesophyll. So by having it deep, it also reduces the amount of sort of airflow of wind moving moisture away from the needle, because this settles the air and you don't get that, rapid wind drying in the winter. Here's an actual cross section of a diagram. You can see the nice amber color of the resin and the resin ducts. These are the recessed stomata. So there's the recess area, and then here's the open chamber. We can look a little closer, and you can see here we have the waxy cuticle, we have the epidermis, and then we have the hypodermis. And then we have guard cells, and we have the pore. And the pore is deep, deep, deep down into the center of the needle. And this reduces the chances of wind pulling moisture from the needle. Okay, so mesophyll review. So we've got spongy mesophyll in the center of the leaf, which have open spaces to allow for gas exchange. We have stomata that open into the spongy mesophyll, whether it's a dicot or a monocot. And then, of course, for dicots, we have palisade mesophyll, which provides structure for the leaf so that it can be extended out horizontally and not collapse. Here's another diagram with the palisade cells, the spongy mesophyll, and the xylem. Remember, xylem is at the top, phloem's at the bottom, because phloem needs to be near where the sugars are being made, right? Summarizing a few things we've learned to date for the differences between monocots and dicots. If you look at the seeds, just by virtue of the name, cot for cotyledon, mono meaning one. So the embryos in the seed have a single cotyledon for monocots and two cotyledons for dicots. I mean, that's got to be the easiest differentiating property. Then there's, like we learned today, there's parallel veins in monocots and reticulated or branching veins or netted veins in dicots. There are no palisade cells in monocots, and there are palisade cells in dicots because, again, the leaves are held out horizontally. Monocots, oh, guess what? This didn't get tripped. This should be reversed. I'll fix that in the slides that you download. Maybe I put it there for a trick. Stomata on one side, that's dicots. Stomata on both sides, that's monocots. Haha. Almost tricked myself. Lastly, when we learned about stems earlier, we talked about how vascular bundles are scattered in the stem of a monocot, and vascular bundles are in a ring in dicots. I got a few review questions. Dicot leaves have stomata primarily where? Lower epidermis. Dicot leaves stomata primarily on the lower epidermis. I'm using the word primarily because the plant kingdom is massive and there are almost always exceptions. The spongy mesophyll's purpose is A, to provide structure in the center of leaf, to allow gas exchange, and for photosynthesis, or photosynthesis takes place here, and D, all of the above. Which one do you think is correct? D, all of the above. So yes, palisade cells provide structure for the leaf, but that doesn't mean spongy mesophyll doesn't either. So maybe that's a bit of a trick question. Water for photosynthesis is transported to the leaf in stomata, phloem, or xylem. Water for photosynthesis. Xylem. Okay, it's a shorter lesson today, but that's fine. It's Thanksgiving weekend, so enjoy. Don't eat too much. Bye.